Hello everyone, welcome back to my channel Study Guide. In the last video we have discussed a little bit of the basics of the PCR reaction. In this video we will discuss more about the PCR reaction, and in this mainly the PCR reaction involves the three processes, that is denaturation, primer annealing and the extension. Okay, and this is the graph that is showing how this process is taking place. Mainly this denaturation process take place at the temperature of 90 to 95 degrees Celsius. At this temperature the genomic DNA that is of double stranded gets denatured into the single stranded. It gets separated out, and so the denaturation step is very important. After this the primer annealing take place, and this primer annealing take place at the temperature of the 50 to 60 degrees Celsius. Okay, this temperature is very important because at this temperature this primers, that is the specific for the template we are using, get incorporated at those sites. After the primer get incorporated at those sites, then the DNTPs can be added further to extend that templates and make our required fragment. Okay, and after this the process of extension take place. The process of extension take place at the temperature of basically of the 70 to 75 degrees Celsius, and this is the third step, that is called the extension. First is the denaturation, second is the primer annealing, and third is the extension. And these are the basic three steps that is required for any kind of the PCR reaction. Along with this the temperature, the requirement of the time is also important. The denaturation take place maximally for the 1 minute, and for the primer annealing we also gives the 1 minute, and for the extension of these fragments we gives the 2 minutes. So this cycle gets repeated many times, and this will help in making the various copies of the required fragment. Okay, and after this there is important things that we should know, that is the primer designing. What is primer? Earlier we have discussed this is the template DNA, and in this template we require some of the precursor so that they get incorporated at these locations and then start the synthesis of the fragment. Okay, and after this we can get our various copy. Okay, and the primer designing is a very important step, because the most importantly for the selective amplification, and the prior DNA sequence information from the target DNA is required. And this is very important, because if we don't know where our primers need to be incorporated, we are not able to get any of the amplified product. So we should know that target template. Okay, template sequence also, there may be ATGC or something ATT. Okay, so according to that we can make the primer so that it can bind at that position and extend that template. Okay, and the design is in such a way that to avoid the non-specific target binding, and we should also take care that the binding should be in such a manner that no other place that the primer get incorporated into the other non-specific position. So there are the certain rules to be followed for making the primer. First is the primer length. And this primer length should not be too short, or it should not be too long. Why not too short? because if it is very short it can hybridize to the non-target site. Okay, this is a fragment and this is our target site, but if our primer is too small it can bind to this region also, this region also, because the sequence can be a same here also. And if it is too long, and this leads to the hybridize at a slower rate. Okay, and the optimal length for the primer is basically of the 80 to 20 nucleotides, and this is the optimal, but this optimal range can be varied according to our experimental condition, but mainly this need to be follow that the primer should be of length of the 18 to 20 nucleotides for the best results. And how the calculation of the length of a primer is done? And by using this formula, that is K equals to G by 2 whole G plus C into 1 minus G by 2 1 plus T. And here the K is the expected frequency of occurrence of the target sequence in a DNA molecules. G, small g, this is the relative GC content of a DNA molecule, and the G, A, T, C, all these G, A are the number of the specific nucleotides in the primers. Okay. After that, the second condition that is required for the primer designing is the nature of the sequence. If there should not be any kind of the inverted repeats or any self-complementary. If there is a inverted repeats or something else, then there is the formation of the hairpin like the structure, which can prevent the primer from the annealing, because if it's formed the hairpin like structure, it cannot bind to the target site. Okay, and self-complementary, in this the hybrid formation will take place and it is not able to bind to the target region on the template. So this need to be considered. Another factor that is important is the TM, that is the melting temperature, and for melting temperature we use the formula. This is known as the Vales Rule. In this, the TM is the TM in the degree Celsius, in which the 4 G plus C plus 2 A plus T. Okay, this is a formula. There is the number of G in our G nucleotide in our primer, then the number of the C nucleotides in our primer, then we calculate the number of A nucleotides and the T nucleotides in our primer length. Then after we multiply it with the 4 with the G plus C and the 2 with the A plus T, and we get the temperature that is for the ideal purpose. And the annealing temperature is typically about the 5 degree below the calculated TM. And also the other things need to be cared in this. The TM value for the two primers should not be differ by greater than 5 degrees Celsius, and also the TM for the amplification products should not be differ from those of the primers by the greater than 10 degrees Celsius. Another factor that is important is the base composition. Okay, we have discussed the three things, and now the fourth thing is the base composition, and there should be a GC content should be between the 40 to 60% with an even distribution of all the 4 nucleotides. So, and these are basically the four methods or the 4 requirement of the for making the proper primers. Now we have discussed how to make our primer and all the rules that is required for making the primer, and in the further videos we will discuss about the how this process is taking place, and the other details will be discussed in the another video. Thank you so much. Please subscribe my channel.